Hey guys, we're doing some cell culture today. We're going to unstick some adherent cells. These are HeLa cervical cells, and then we're going to count them and split them and maybe plate some. So first thing we want to do is remove our old media. Since these are adherent cells, we can use an aspirator if you so choose, or you can use a pipette in a waste jar. I'm doing 2 flasks at a time, but 1 flask or multiple is good if you can handle it. You want to be careful not to scrape the cells off the bottom when you're taking out the old media. And then we have our warm trypsin and our warm PBS ready. Trypsin does not like to work with media, and so it's best if we rinse the cells with PBS first. These are 275 flasks, so they do 10 mLs of media when they're in culture, and then for PBS we can just do 3 to 5 mLs. You just kind of put it in there, give it a little zhuzh, and rinse it out. If you have floating cells, use a pipette and then collect everything into a collection tube to spin down. If your cells are super sticky, try rinsing them with ice cold PBS at 4 degrees first instead of warm PBS. And that might help your cells unstick. Besides trypsin, we can use Accutase, and then there are also some non-enzymatic forms of cleaving those cell adhesion bonds. So we have our rinsed adherent cells in the flask, and so now it's time to trypsin. And remember, anything you put in the hood should be sterile. And try to keep things off of the vent. And so for a T75 flask, I use 3 mLs of trypsin. These 2 flasks are identical, so I don't mind using the same pipette for this step, but if your flasks are different, make sure to keep that in mind. And now we're going to let these incubate with the trypsin for about 5 to 10 minutes at 37 degrees in your incubator.